Right, let's dive right in. Basic bacteriology for step one. I get it. This topic is an absolute monster. But look, we're not going to try to boil the ocean today. This is a high yield blitz designed to give you the absolute must knows, the mnemonics, and the clinical tie ins that'll help you crush these questions on exam day. So here's our game plan. First, we're going to start with the fundamental blueprint of the bacterial cell. Then we'll talk about how we actually identify these bugs in the lab. After that, and this is where the real high-yield stuff is, we'll get into their survival strategies and virulence factors. Finally, we'll wrap it all up with a quick-fire quiz to really lock these concepts in. Sound good? Let's do it. First up, the bacterial cell itself. And you've really got to nail this down, because if you don't get the basic architecture, you can't understand how antibiotics work, how our immune system fights back, or what makes these little guys so dangerous. This is the foundation for everything else. Now, this slide right here shows probably the single most important distinction in all of bacteriology: gram-positive versus gram-negative. It all comes down to the cell wall. On the left, you've got that thick, beefy, purple-staining wall of a gram-positive bug. And on the right, you see the more complex, multi-layered setup of a gram-negative. Okay, so let's break this down. For gram-positives, the two things to remember are 1, a super thick peptidoglycan layer, and 2, lipoteichoic acid. That lipoteichoic acid is a major virulence factor that kicks off a huge inflammatory response by inducing TNF-alpha and IL-1. Now, for gram-negatives, it's a totally different story. They have a thin peptidoglycan layer, but the real star of the show is that outer membrane. That's where you find endotoxin, also known as LPS. This is the big bad guy that triggers that massive inflammatory cascade leading to septic shock. Let's hit these other structures real quick. Capsules are basically these slimy sugar coats that make bacteria hard for our immune cells to grab onto and eat, a huge virulence factor we'll circle back to. Spores, think of them as tiny bacterial survival pods. They let bugs like Clostridium and Bacillus survive almost anything, heat, chemicals, you name it. And finally, you have pili, which are like little grappling hooks for sticking to surfaces, like your bladder wall, and for swapping DNA, which is a huge deal for spreading antibiotic resistance. Okay, so we know what they're made of. Now, how do we actually ID these guys in a real clinical setting? Well, this is all about stains and some very special cultures. And boom, here are our two most important stains, visually. On the left, that's your classic gram stain. See those pink rods? That screams gram-negative. And on the right, that bright red is the telltale sign of an acid-fast stain. This is what you need to see bugs like Mycobacterium tuberculosis that just won't play ball with a regular gram stain. This table is pure gold for your studying. You absolutely, positively need to have these associations down cold. Giemsa stain, that's for those tricky intracellular bugs like chlamydia or parasites like Plasmodium. Ziehl-Neelsen is your go-to for mycobacteria that high mycolic acid content in their walls makes them acid-fast. And if you see a question mention India ink, your brain should immediately jump to Cryptococcus neoformans. The ink stains the background, leaving this beautiful clear halo around its thick capsule. Just like stains, some bacteria are super picky eaters. They need special media to grow, and the USMLE loves to test these pairings. H. influenzae on chocolate agar, Neisseria on Theramartin. These are classics. McConkey agar is great because it helps us tell if a bug ferments lactose. If it does, the colonies turn pink. And maybe the most famous of all, E. coli on EMB agar. It produces this unmistakable, iridescent green metallic sheen. You see that, you think E. coli, period. It's an easy point on exam day. All right, let's level up. We're moving into bacterial survival strategies. And let me tell you, grouping bacteria by how they survive is incredibly high yield because these categories tie directly into the diseases they cause and who they infect. So remember that capsule we talked about? Here's why it is so clinically important. This polysaccharide coat is basically an invisibility cloak that helps bacteria evade our immune system. So what happens if a patient doesn't have a spleen, the main organ that filters these bugs out of the blood? Well, they're at a huge risk for overwhelming sepsis. This is a critical clinical link you have to know. And here it is, the mnemonic you absolutely must commit to memory. Please shine my skis. This covers your big encapsulated bacteria: Pseudomonas, Strep pneumo, H. influenza, Neisseria, Salmonella, Klebsiella, and Group B strep. Honestly, the big three to always have top of mind are Strep pneumo, H. flu, and Neisseria meningitis. When you see a question about a patient who is asplenic or has sickle cell, this list should immediately come to mind.
Another key group, the urease-positive organisms. You can remember them with the mnemonic P chunks. These bugs produce an enzyme called urease, which jacks up the local pH, making things more alkaline. The classic board question here involves Proteus mirabilis. It causes UTIs, and by making the urine more alkaline, it helps form these massive struvite kidney stones. You might have heard them called staghorn calculi.
All right, now for the bacterial weapons of war, virulence factors. These are the specific tools, the enzymes, the toxins, that let bacteria cause disease. Step 1 loves to ask, what's the mechanism questions, and the answer is very often one of these factors. So let's start with a clinical scenario. You've got a patient with chronic granulomatosis disease, or CGD, and they keep getting these awful recurrent infections. We know their immune system is weak, but what's the specific biochemical reason they just can't handle certain bacteria? The answer is all about one enzyme, catalase. See, patients with CGD have a defective NAPDH oxidase, so their immune cells can't make that respiratory burst needed to kill bacteria. But catalase-positive organisms can take the little bit of hydrogen peroxide the patient can make and just neutralize it. They basically bring their own fire extinguisher to the fight. Those bubbles you see? That's pure oxygen, a dead giveaway for a positive test. So, which bugs do you have to worry about in a CGD patient? You need to know your catalase-positive organisms. The mnemonic, notoriously big bubbling hassle, can help you remember them. It includes bugs like nocardia, staphylococcus, and listeria. And for your exam, the absolute classic example is Staphylococcus aureus causing abscesses in these patients. And finally, let's nail down some pigment producers. These color buzzwords can be just slam dunk easy points. You see yellow sulfur granules draining from a jaw, think actinomyces. Remember, Israel has yellow sand. A burn wound with a blue-green pigment and a fruity grape-like smell, that's gotta be Pseudomonas aeruginosa. Think arugula is green. And if a question mentions red pigment, especially in a catheterized patient with a UTI, think serratia, like red sriracha. You need to know these color associations cold. Okay, take a deep breath. You've just downloaded a ton of high-yield info. Now it's time to put it to the test and really make it stick. Let's run through a couple of USMLE-style questions.
First question, a young boy with CGD. You see that diagnosis in the question stem, and your brain should immediately scream one specific enzyme. What is it?
Yep, the answer is B, catalase. It's that classic connection we just talked about. CGD means no respiratory burst, and catalase-positive bugs can just shrug off the patient's weakened immune response. This is a bread-and-butter immunomicrobiome-integration question.
All right, question 2. A patient has had their spleen removed. This is another one of those classic clinical tie-ins. What's the number one category of bacteria we are worried about in this person?
The answer is, of course, C, encapsulated bacteria. Remember, the spleen is like the bouncer of the bloodstream, responsible for clearing out opsonized bacteria. Without that bouncer, the encapsulated bugs from our Shine Skizz list can throw a deadly party. Let's end with one final thought to bring this all home. When you're working up an infection and you're faced with an unknown bug, what's that single, fundamental, first-line test that tells you more than anything else about its structure and how it might behave? It's the Gram stain. It all comes back to that first slide, to that fundamental difference in the cell wall. If you can master that, you've mastered the foundation of all of bacteriology.